From 2016 to question number 14, which one of the following mechanisms is used to coordinate the expression of multiple related genes in eukaryotic cell? So, which one of the following mechanisms is used to coordinate the expression of multiple related genes in eukaryotic cell? So, what is that? So, normally in prokaryotic cells, you have seen the presence of operons like galactosoperon or lactosoperon. That is the most common operon. What happens is that there is a single promoter, okay, and the single promoter drives the transcription of all the genes that are required in the that pathway. Like you have A, B, C, D, those kinds of genes. So they are if the transcription is turned on, then all these genes will be transcribed. So these are related and they are kept in a single operon. Okay, this happens in the prokaryotes. Okay, but in eukaryote, the operon system is not there. But still, there are many genes which have to be upregulated at a single time if there is a environmental signal or stress signal. So, uh, how do the eukaryotes they regulate the expression of multiple related genes? What is the mechanism? The options are: first, environmental signals enter the cell and bind directly to the promoters. This is one option. Genes share a common intragenic sequence that allows several activators to turn on their transcription regardless of their location. This is the second option, having a common intragenic sequence. Now, see, genes are organized into large operons, allowing them to be transcribed into a single unit. So, this option is wrong because we know that operons does not exist in eukaryotic cells. So, this is wrong. D. Genes are organized into clusters with local chromatin structures influencing the expression of all the clusters gene at once. So this is the last option. So which one is the correct option? Which is the one mechanism which we use? So this is from a paper which shows that uh, so first of all operon which does not happen in eukaryote. So that is to completely neglected. You can see here like in bacteria we have lac Z, lac Y, lac A all together they are transcribed under a single promoter. So this is how like related genes are transcribed in prokaryotes. Now this is another kind of gene system. Now in eukaryotes what we see is gene clusters. What is that? 3D transcription condensation. What see? Several genes, gene 1, 2, 3, 4, all are present on different chromatins. Okay, they are present on different chromatins, but they are they cluster together such that like if you have heard about epigenetics, then you know that acetylation or methylation of the chromatin can lead to different structures like heterochromatin or euchromatin. So, if they are clustered together in a single region, then normally the enzymes for acetylation, methylation, all those things will be present. Like here, if we see, if they are present in the same pool. So, the if there is an environmental signal, then this different kinds of regulators can be activated and they can act on all these genes together because they are in the similar vicinity. So this is how the eukaryotes uh, control the expression of multiple related genes in the eukaryotic signal. So this is the thing. They are organized into clusters with the local chromatin structures influencing the expression of all the clusters gene into at once. Okay, and not I'm neglecting these options because the second option, genes share a common intergenic sequence and allow several activators to turn on their transcription. So genes activators normally do not occur inside an intergenic sequence. They are several bases upwards or downstream of the gene. Okay, if we have a gene over here, then the activator will bind several bases upstream or downstream. It will not bind to any intragenic region. So I am neglecting these options. And environmental signals enter the cell and bind directly to the promoters. This is this can happen to some extent, but this is not actually the case. Okay, so the option D is the right answer.